The ear is one of the primary sensory organs in humans. It allows us to listen to language, as well as listen to music, and also sense the auditory world around us. But how does the ear work? In this video essay, we will discuss the inner workings of the ear, as well as demonstrate how these neurons work within Python using a single-layer Leaky Integrate-and-Fire feed-forward network.
The human ear receives input from the natural world as changes in air pressure, which are sound waves traveling through the air. These changes eventually reach the outer ear, which acts as a sort of cone that funnels the sound waves into the inner ear. The first step is converting the input into neuronal activity in the eardrum, which vibrates because of these changes in air pressure. These vibrations are transmitted through a chain of 3 tiny bones in the middle ear known as the ossicles: the hammer, the anvil, and the sternum. And it travels towards the stapes, which then push against the oval window of the cochlea, further amplifying the vibrations. Because of the mechanical movement of this oval window, the fluid contained in the cochlea begins to vibrate, initiating a response in this fluid-filled structure. Inside the cochlea, the basilar membrane vibrates at specific locations, depending on the frequency of the sound. It is its spiral shape that allows it to be sensitive to different frequencies. So high frequencies are affecting one end and lower frequencies are affecting the other. This is where the conversion into electrical signals happens. There are 3 fluid-filled compartments containing different specialized hair cells. The type we focused on primarily are the inner hair cells, which bend due to the movement of the fluid, causing them to finally release neurotransmitters to adjacent auditory nerves.
First, we have to convert an audio signal into input for our program. To do so, we opted for the Librosa library in Python, which is a versatile library used to analyze incoming signals, particularly audio. We use it to import the audio as well as its powerful spectral analysis capabilities, particularly the fast Fourier transformation and the short-term Fourier transformation, which is in discrete time intervals. To model the function of the basal membrane, we first had to find a way to filter the incoming audio signal into discrete frequency bands. After importing the audio into Python as a NumPy array using Librosa, we performed a discrete short-term Fourier transformation. Last, we had 2 variables, which were 2D NumPy arrays, each one representing the magnitude spectrogram of the left and right audio channels, respectively. The output being formed by 2 variables, where the first one corresponds to the number of frequency beans, and the second one corresponding to the number of time windows the signal has been divided into, determined by the length of the audio file. As an interpretation, the rows represent frequency bins, showing the magnitude of each frequency component present in the signal, whilst columns represent the time frames, indicating how the frequency content changes over time. The values in the array being the magnitudes of the frequency components in each time frame. By doing so, each band of the Fourier transformation can function as the receptive field for a particular inner hair cell. By fitting the data of each band into a LIF neuron, we can simulate the spiking behavior of the individual neurons and their respective receptive fields. The number of bands also becomes the number of neurons per year.
This code simulates how audio signals can be processed and interpreted as neural responses. The first part of the code sets up a simple graphical interface using tkinter, allowing the user to select an audio file. Once the file is chosen, the import audio function captures its path and the GUI window closes. Next, the load audio function is called to read the selected audio file. Librosa is used to load the audio as a stereo signal, returning the audio data array, the sample rate, and the length of the audio in milliseconds. The code then performs a short-time Fourier transformation on both the left and right audio channels through the compute-FFT function. This operation splits the signal into overlapping time windows and calculates the frequency spectrum for each window. The result is a 2-dimensional array representing the magnitude of the signal's frequency components over time. A parameters function sets up default parameters for the Leaky Integrate-and-Fire neuron model, including properties like membrane potential, thresholds, reset potential, and time constants. The main part of the neural simulation is handled by the LIF neuron function. This function calculates the membrane potential over time for each simulated neuron, recording spikes for when the potential crosses a defined threshold. The function returns both the membrane potential trace and the times when spikes occur. The simulate neuron spikes function processes each frequency band of the short-time frame Fourier transformation data. It scales the input current for each band and ensures its length matches the simulation time. This scaled input is then passed to the LIF neuron function, which simulates spiking activity and records the spike times for each band. Finally, the plot raster function visualizes the results. It plots a raster of spike times for both the left and right audio channels, showing how each frequency band activates over time. Run cochlear model orchestrates the entire process. It begins by loading the selected audio file, then computes the short time frame Fourier transformation, sets up the Leaky Integrate-and-Fire neuron parameters, simulates the spiking activity for both audio channels, and then displays the raster plot of neural spikes.
Now, what are some ways in which this model could be used in research? Firstly, the output of the neurons from our model can effectively be connected to any neural network structure. For instance, a network using Hebbian learning may be able to store the neuronal data as memories. Firstly, the output of the neurons from our model can effectively be connected to any neural network structure. For instance, a network using Hebbian learning may be able to store the neuronal data as memories, or even generate entirely new music. Furthermore, researchers could use this neuronal model in reverse to decode nerve data into their respective spectral data. There are potential uses in development of auditory prosthetics, neuro-inspired computing and machine learning, psychophysics and perception studies, and much more.
Some advantages of using this code are its customizable parameters, such as the LIF neuron model and the STFT computation parameters, which can be adjusted to simulate different scenarios and hypotheses. Also, it allows integration with real data, as the code can be modified to take real-world audio samples, making it useful for applied research. It allows for visualization, as the code includes visual tools like the raster plot, which helps researchers intuitively interpret the spiking activity of neurons in response to sound. Some of the limitations would be the fact that it's a simplified neuronal model by using the LIF model. Moreover, the computational intensity is pretty high, as it does not have enough resources to process high-resolution audio or large number of neural simulations. And finally, it does not allow for large-scale generalization, as certain complex real-world auditory processing tasks require much more sophisticated models or techniques.